Hi, everybody. It's been a long time since you've heard my voice probably, and we're going to go ahead and start medicines and drugs, option D. This is going to be our part one. So we're going to have some definitions today. Ultimately, we're gathered to listen to the video, take some notes as we go along. Um, you're going to have some good news here, and that is that this part of the learning is all going to be more discussion based and less about taking tests and more about sharing information and discussing those. So let's go ahead and get started. Pharmaceutical products. So the first thing we have to do is be able to discern between a medicine or a drug, and what all these things are. So a chemical that does one of more of the following. So we're going to be altering physiological state, um, encountering sensory sensations, moods, or emotions. So pharmaceutical products are used for a lot of things. Um, when we talk about development of a drug, and this is going to be really important to us as of right now because development of a drug is something we badly need to happen. So first we have to identify a disease. It could be a new disease. In this case, we have a new virus. Identify the target or gene or enzyme which is necessary for the disease to progress. This is part of what we are trying to do right now. And then um, what we're going to try to ultimately do is find a molecule that can act on that gene or enzyme that is part of the body that is going wrong. We might have to modify a molecule to get it to do that. All right. So our next two terms that we need to pay attention to include ways in which we describe preclinical trials. Um, this is where we are going to be working on the development of this drug as in the lead molecule to attack the drug. And the two ways you can do this is in vitro, um, where it's actually tested on animal or human cells that have been removed from the body. So now they're in a petri dish in vitro. You've heard of in vitro fertilization. That means that we are doing this to cells which have been removed from the mother's body. So the mother's egg, in other words. In vivo is testing on live things. And this is the kind of thing that has to happen before we can put it out there to the general public. And so this is defined as testing on live animals, usually at least three different species, to establish a lethal dose. And this is where 50% of the subjects are killed. And so this is where we are using animals instead of people because we don't want that to happen to the people in our study. Okay, so what we're referring to is something called the therapeutic index. The therapeutic index is a way to compare the effect, as in the helpful effect of the drug versus the toxic effect of the drug. And so you can see in this graph we have two lines, one representing where 50% of the uh, people are given the therapy or experience the therapeutic effect, and then another line that shows where 50% of people experience a toxic effect. And so what we're going to be looking for is broad range or a broad area between these. In other words, we'd like these two graphs to be as far apart from each other as possible to have the greatest therapeutic, as you're going to see, window that can lead to drugs being taken that will not harm people, but that will produce the greatest amount of therapy or positive results. Okay, so in all times we're going to be comparing these drugs effectiveness versus their safety. They can be very effective, but if it is way too easy to overdose, then and experience a toxic effect, then we don't want that to happen. So when we're comparing, okay, when we use it in animals and when we use it in humans, in animals, as we've already said, we're going to compare the lethal dose versus the effective dose. 50% of each are experiencing that. And then we're going to compare the toxic dose, in other words, where the dose becomes toxic to humans as compared to its effective dose. In other words, the effectiveness of the therapeutic effect. Okay, So the greater the index, this is simply referring to the lines being separated from each other, the more chance we have to be able um, to experience good results. Okay. Uh, let's see. What about these over-the-counter drugs and being able to understand what is an over-the-counter drug and why are we allowed to even take any drugs over the counter? Shouldn't all drugs be monitored by our healthcare providers? So let's take a look at that. Over-the-counter drugs are drugs that are defined as safe and effective for use by the general public. In other words, the general public could take this drug without too much worry that what they're taking is going to cause them a bad result, so a toxic result. So if we look at our window here, the therapeutic window, that is starting with about a concentration of pretty much a 10% effect of therapeutic effect on people, all the way out to about 90% of people are experiencing a therapeutic effect, and then, if you'll notice, it starts to experience some form of toxicity. So this is why you'll notice that in some medications, some people do really well with medications and experience therapeutic effects. And then other people, like me, when I take over-the-counter medications for cold, I usually don't experience this therapeutic effect like other people do. Like if I were to take NyQuil, it does not put me to sleep. It does not help my symptoms. So that's something that I don't experience the therapeutic window very well. So it's not very effective for me. If I were to take more, I might end up in this high toxicity range, and I wouldn't want that. Okay? So if you'll notice is how can we classify a prescription medication into a over-the-counter medication. Okay? So the results or the indications should be similar. So which one uses the prescription and which one uses the OTC. In other words, we want to be able to recognize, oh, I have a sniffy nose or a stuffy nose. I should be able to take this medication. I don't actually need a doctor to tell me. It's a pretty easy way for me to diagnose myself. A favorable um, drug interaction. In other words, it doesn't highly interact with other drugs which I might already be prescribed or taking. And um, in other words, it should not have a narrow therapeutic index. So you'll notice this distance between the two lines. That therapeutic index, we want this number from, and this one goes from about 13 to about 28. We want this range to be as high as possible to limit the amount of possible overdoses that might occur. Okay, so let's go on to, well, what about other medications? Oftentimes when a drug is being used, we take into account the placebo effect. In other words, can a patient understand that they are receiving a drug and that it's going to help them? And might the patient actually experience the effect without actually having had the drug? So what is the placebo effect? This occurs when a person experiences a positive therapeutic effect because they believe themselves to be given a drug that they don't know is not a medication, but it's really just like a sugar pill, something that will not harm them. So you'll see some of the percentages of 48% of doctors prescribe placebos at least 10 times. 16% of the participants in a Parkinson's disease trial showed improvement. 85% of the reduction in cough medication trials are due to the placebo effect. Wow, that's amazing. With 15% due to the active ingredient. In other words, if we just take some cough medicine that we think is going to help us, we 85% of the time are going to experience results, and that cough medicine might actually not have any medication in it whatsoever. So, interesting to know. Um, how would you allow for the placebo effect in carrying out clinical trials? In other words, what part of the placebo effect is that going to be in a clinical trial? And we're going to get down to talking today a lot about clinical trials. How do you analyze those results? And is it ethical for some people to receive the medication and other people to not receive the medication? So let's go on and talk about that. So in drug trials, specifically we mean on humans, and this is where we're going with having to prepare ourselves to um, get the vaccinations ready to go for all the people that are going to, and that's all of us, by the way, that are going to need this vaccination, I'm sorry. We have to test its effectiveness, and we have to use the placebo effect to do that. So this is a blind trial. Now that's another term that we need to make sure we know. In other words, you could possibly, being in this trial, be one of the patients who received the placebo. In other words, no actual medication will be given to you, but you don't know that. You could also be one of the people that is given the actual active ingredients in the drug. You don't know that either. So um, one half of the group of participants receives the sugar pill, one half receives the actual medication, and that way they are able to have a control without the knowledge of the person knowing what you know about the placebo effect. In other words, you can receive a medication thinking that you're going to get better because you receive the medication, even if what you have is a sugar pill. They wanted to eliminate that part of the effect of the medication. So they want to be able to study what the actual effect of the medication is, and not, in other words, taking out what the effect of the placebo. In other words, in this example, with 50-50, all patients should expect to or could experience the placebo effect and might not know that. In fact, probably will never know that. Our picture here is we have the amount of people that we have to notice phase 1 through phase 4. I am imagining that they are going to speed this process in clinical trials up in a major way for us to be able to have enough of a vaccine that is prepared for us, but they're going to have to speed it up quickly, and this process has already begun. So my understanding right now is that we are somewhere between phase 2 and phase 3 right now, and that with these number of participants being significantly higher than we would normally expect in the clinical trial process. All right. So virus drug testing. Why does it take so long? So I put this little thing here up, and I'm going to move my picture over to the side here because I think that it's getting in the way. So why is it taking so long? So there we go. You might notice that in a typical process, and this was a process for treating the Ebola virus, and so being able to work on the cells, being able to separate things out, and evaluating which compounds are working, which compounds are not. So this works you through a step-by-step process of what exactly had to be done to start to create some kind of Ebola virus vaccine. And so that's what this process is, and that's a pretty long process. Um, so if you click on this link right here, this will take you to a website that is talking about one of the companies out there that is starting with clinical trials on working on a vaccine. And so that gives you a little bit of background information on one of those companies. You should click on that link and follow that and just find out a little bit about one of those companies whose all of these companies are beginning clinical trials right away. Notice what it says. It would still take about 3 more months to conduct the first stage of human testing. First stage of human testing. That was 10 to 80 people. And so this is a long process, and another 8 months or so to complete the next stage. So it's going to take a long time. What about new vaccines? Okay, so they're going to require a lot of research. And so I'm going to click ourselves onto the next slide. How does the vaccine work? This is a link to the New York Times, the Los Angeles Times article, but I want you to read through. How does the vaccine work? Our questioning on Friday is going to be based on this Los Angeles Times article. How does the vaccine work? And we are not going to go on to this part. I'm going to click us back here. So we're stopping right here. How does the vaccine work? And this is the basic beginning of that video. And our question on Friday, or questions on Friday, and conversation will be based on this article and some of the other. If you come to a link in the article, please go and check those out as well. All right, everybody. Thank you so much, and I will talk to you on Friday.